Hello, everyone. My name is Vanessa Fiore, and I'm here to present my study on the effectiveness of anthropogenic debris removal by the Seabin system within the Ottawa River. My supervisors include Dr. Jessie Vermeer and Dr. Scott Finley. It is very well known within the scientific community and around the globe that plastic pollution poses a pervasive and ubiquitous threat to aquatic ecosystems, impacting fauna, natural habitats, and humans alike. In response, numerous innovative companies have been developing products, services, and technologies to help clean plastic pollution and anthropogenic debris from aquatic ecosystems. One such innovation includes the Seabin. It is a bucket-like contraption that collects floating debris via an inlet and a catch bag. Recently, the Nepean Sailing Club has acquired a Seabin as part of a pilot project in collaboration with a local organization called Deep Clean Blue. And the purpose of this study is to assess the Seabin's potential in cleaning debris from the Ottawa River. This is being done through three experimental objectives. The first is looking at the content that gets collected. Second is evaluating the relationships between the variables of the content that were identified, as well as investigating the potential ecological trade-off. And then third is to conduct future projections and see how much debris can be collected from the river over the course of a year. For the methods, we started by collecting the samples from the Nepean Sailing Club. So they would collect samples over a period of 24 hours, meaning the Seabin for an entire day would be collecting content, and we'd come and get that content and bring it back to the apartment for analysis. So once we collected it, then we would separate the content based on plants, anthropogenic debris, and animals, and we would weigh each type of content from each sample. For the plastics, we would count the number of plastics as well as identify the type and size it. And then for animals, we would look at the species as well as the number of individuals captured within each sample. For the statistical analysis, most of the observations were recorded on Excel, and those same observations were visualized. However, RStudio was used for the bulk of the data analysis, including simple linear regression, multiple linear regression, and temporal autocorrelation. According to our results, the average weight of anthropogenic debris amongst samples was 22 grams, plants was 740 grams, and animals was 8.75 grams. It is interesting to note that the plants do have the highest quantity within the content, and this trend will be explained in the following slide. However, for now, we'll focus on the anthropogenic debris. So here in this graph, we compared the plastics versus the non-plastics and the types within each category. And then we also compared here the total number of plastics versus non-plastics. So accordingly, there is more types of plastics that have been collected. And the most common type of all the anthropogenic debris is plastic fragments. This comes as no surprise given that plastics that remain within the environment for a period of time, for an extended period of time, will eventually be subjected to mechanical forces and UV rays and eventually break down into smaller pieces. So this is what is likely being collected by the Seabin. And then there is, once again, a higher quantity of plastic debris that gets collected versus non-plastics. So as previously mentioned, we did want to compare the relationship between the debris, the number of pieces of debris, and the weight of plants. We also did this for weight of debris and weight of plants. And we found that there is a positive correlation that exists between the debris and the plants, which indicates that as we collect more plants, more debris also gets collected. And this is due to entanglement. So much of the debris will become entangled with the floating plant matter, which is, again, advantageous and represents a symbiotic relationship. And we also compared our results to that of U of T and found a almost identical slope, which was really exciting for us, as this shows that these conditions and this trend can be replicated. So we also looked at the species collected, and we found that the most common was Dinerchus, followed by snail shells, other type of biological matter, and western banded killifish. There were no species at risk present, which is a good sign that the Seabin doesn't pose a risk to various species. However, there was a couple of live western banded killifish, so they're smaller species and should be like the Seabin should exercise caution when operating. In terms of future projections, around 2,928 grams to 7,771 grams are projected to be collected by the Seabin throughout an entire year. These are the stats for the pieces. And for the temporal autocorrelation, there is none that exist, as you can see here. And this indicates that subsequent temperature events do not affect what gets collected by the CBIN. And finally, our conclusion is given the low ecological trade-off based on our qualitative analysis, as well as the quantity of plastic that gets collected and the potential of what it could become in a year, the CBIN is a potential solution to help clean plastic from the Ottawa River. Thank you so much, and have a great day.